So this is a study that we perform again in collaboration with Heidelberg University, and it's well known and Mark Raab in particular, and was led by Anthony Cirincione from our lab, and he will present the data. And it's kind of very interesting because looking at the hyperdiploid myeloma, which is probably one of the most frequent events in myeloma that we know that half of myeloma have hyperdiploidy as probably initiating events, and that's what everybody is using in their slides, in their books, in their review. Hyperdiploidy is initiating event, which means it's the first event in the myeloma life history, is where myeloma is born after hyperdiploid. Now, one of the data we're going to show is that in some patients, around 10%, we have evidence of alterations that precede hyperdiploid. So hyperdiploid is early, but it's not necessarily the first event. So that's one point. The second point is that we found a new mechanism of gene dysregulation in multiple myeloma that can also be applied to other tumors, because I think it's a new concept. So when you have a trisomy or any gains, you have 3 copies. And when you have a deletion on the gain, you still have 2 copies, because you go from 3 to 2. And so the question is, are these random deletion or do they have a role? Because the other 2 copies should sustain the expression of whatever is lost. So it turns out that these events are very common. They are associated with gain of function events, joining a gene with a regulator's distal dysregulator, or they also inactivate tumor suppressor gene. And the way in which this happens is through the inactivation of the other 2 remaining alleles through cross-transcriptional mechanism, and we have this data from single-cell ATAC, epigenetic mechanism that probably suppress the other 2 genes. And so even if you see 2 copies, even if it seems to be a diploid section, it's actually a deletion. And that can only be captured by whole genome sequencing, because other technology would miss and you would not be able to understand if this diploid region is a deletion or not. So that's again prioritize the importance of whole genome sequencing and highlight the complexity of multiple myeloma and how many events we haven't yet figured it out.